I think we should probably kick things off.
Yep.
Okay, so I just want to start by orienting everyone to this virtual room. So you'll see that we have a few slides that we're going to go through during the first 15 or so minutes of this session. If you want to see those more clearly, you can double click on that window to enlarge the slides, and then double click to minimize when we're ready to discuss. But Allie, do you want to start with some words about mini PCR and BioBits, and then we'll introduce ourselves? Yeah,
for sure. So I'm Dr. Allie Huang. I co-developed BioBits, which I'll explain a little later in the slides, with Jess here when we were both in graduate school. So I did my PhD at MIT, and upon graduation and working on this idea of applying synthetic biology to educational purposes, I went and joined Mini PCR Bio, which is a small biotech company located near Boston, which focuses on making molecular biology and synthetic biology more accessible to everyone. And we do that through really accessible equipment as well as really innovative curriculum. We can talk a little bit more about that in a bit.
Great. I'll introduce myself. My name is Jessica Stark. I also have a PhD from Northwestern in chemical engineering. Before that, did my undergrad at Cornell, also in chemical engineering. And now I'm a postdoc at Stanford working in chemistry and immunology. And in my PhD, as Ali mentioned, we co-developed BioBits kits, which we hope are going to provide some solutions for hands-on biology education, both during the pandemic, but also increased accessibility to those kinds of experiments under normal circumstances. I want to welcome everyone to put their comments and questions in the chat as we go along. Like I said, we're going to have a short presentation, and then we're going to open it up to a broader discussion. So get your questions ready, get your thinking hats on, and I hope that we can have some good discussion later in this session.
Yeah, let's get kids excited about science.
For sure. Okay, so to kick things off, I just want to start by talking about the need to increase scientific literacy, because this remains a pretty significant challenge in the United States. A 2015 study showed that citizens and scientists disagree on really key scientific issues, everything from whether it's safe to eat genetically modified foods, to whether evolution is fact or fiction, to the importance and safety of childhood vaccines. And this is underscored, kind of a key reason for this discrepancy is underscored by this poll that showed that scientists believe that increased K-12 STEM could be a main way to combat scientific illiteracy. And overall, this challenge and need to increase scientific literacy in the public is important, because as we've seen in the past few months, if the broader public doesn't understand scientific issues, that leaves them vulnerable to misinformation from the press and other outlets, and really has led to continuing misconceptions about the COVID-19 pandemic that is really inhibiting our ability to combat this effectively. So to drive home really how we can address this problem, I want to highlight on the next slide.
Yep, I'm trying.
Sorry.
It's not letting me advance. I've been clicking for a while.
There are, so sometimes I have trouble with this too. There's arrow keys. If you move to the bottom of your PowerPoint screen, the bottom left-hand corner, sometimes you get the arrow keys that show up. That can help advance.
Next. Yeah, I think I'm just gonna, my computer did not like that.
Okay, so let's try again.
Let's try again. I think my PowerPoint itself, I need to restart PowerPoint, it crashed.
No problem, it happens.
I am actually going to link, yeah, my PowerPoint just crashed. I'm going to link Google Slides version of our slides. Oh yeah, so the data we got, it was a AAAS study that they did in 2015. Someone just asked that in the chat.
I'm actually I have a link from Pew Research that I'm gonna put in the chat about where you can find more on this data.
Yeah, sorry, my PowerPoint completely crashed. So I linked the Google Slides so people can follow along in case this doesn't work out. All right. Yeah, everything's frozen on my side.
Why don't I try to share?
Okay. All right, I got to back up if we wanted to.
Okay, do you want to try sharing again? Otherwise, yeah. I'll take a turn.
Sorry, guys. This is the reality of the Zoom days, I guess. All right, here we go.
All right, we're back in business. So in order to combat scientific literacy, scientists have identified K-12 STEM education as a key mechanism by which we can improve students and the public's understanding of science. And there is still a need to create more inclusive avenues for STEM education. A study from the NSF in 2012 found that 20% of high school students fail to take a science class before 11th grade. And so there really still exists a need to create more inclusive opportunities. And especially in light of current events, socioeconomic status as well as racial and ethnic backgrounds, we know influence barriers that students may face in terms of their ability to pursue careers in science. And unfortunately, these same backgrounds and communities are disproportionately affected by the COVID-19 pandemic. So all of this to say that, especially in light of the current climate, it's really on us as educators to come up with effective and inclusive ways to teach STEM. Hands-on activities have emerged as one of these ways to foster inclusion and improve performance in STEM disciplines. Hands-on activities have been shown to improve both in student engagement and educational outcomes in STEM. And as a result, there's been a surge of development of hands-on kits for teaching subjects like physics, chemistry, electronics, programming, engineering. However, the pace of development of these sorts of hands-on science kits, particularly for biology education, has lagged behind. And we'd like to argue that that's likely due to challenges associated with growing cells. So as many of us appreciate, cells are the basic unit of biology, and so many biology labs require growing and storing cells. And that requires significant equipment and expertise in laboratory processes. So for example, let's consider a typical protein expression experiment like the Biorad pGlow lab. This would mean that you need to transform bacteria with DNA, select those that express your protein of interest, in this case, a fluorescent protein. And to do that, you might use something like a UV transilluminator to figure out which of your cells are fluorescent. That's an expensive piece of equipment. You then need to grow your cells up to make enough protein to then purify and observe. And to do that, you need large shaking incubators, which again are costly and not very portable. To purify your protein out of those cells that you've grown, you need some kind of purification setup, whether that be an automated chromatography system or a more basic kind of gravity flow system, but still requires resources as well as specific scientific know-how. And then finally, you can isolate your protein and observe it. But you may, in the spirit of teaching students how to be rigorous scientists, you may want to quantify how much protein you've made, for which, and for this activity, you would need something like an expensive spectrophotometer. So I've tried to highlight as I went along here, but you can appreciate that each of these steps requires significant expertise and usually an expensive piece of equipment, which makes this really challenging for many educators to implement in their own classrooms and really impossible under the current circumstances where we're trying to implement hands-on activities at home. So I'm going to hand things over to Allie now to talk about how we might address this problem.
Yeah, and just as an aside, this is not a knock to any of the other existing molecular biology activities that are out there for classrooms right now. If the teacher and the school is able to get those for their students, it's great for them to be able to grow cells and use that, because if they go into research, that's what they're going to be doing. But the reality of it is that not all schools are going to have access to something like that. And especially in, this day and age where we have to do a lot of learning and teaching at home, I'm sure a lot of parents aren't too crazy about the idea of their students culturing E. coli cells in their bedrooms, although that does sound pretty fun. But so we wanted to offer up some potential solutions that we've identified to help with this issue, and then from there kind of open it up to discussion about, what the pros and cons of these solutions are, how do we implement these solutions, who are the key stakeholders and players we need to talk to in order to get these solutions implemented. So, the first one that comes to mind is the idea of virtual simulations. And this is a shout out to Todd and Sandy from Molecule World. So, their app is a really great example of a kind of a virtual way to be able to examine proteins. So their app allows you to pull protein sequences from like the protein data bank, and you put it into your app, and then you can highlight different residues and spin it all around and like really kind of see like what this protein looks like from like a 3D perspective. And there are also similar simulations and virtual labs out there where students can simulate running a PCR or simulate running a gel using a just like a computer program or some sort of computer app. And this way, the pros of this is that all the student needs is a computer, and you don't really need any of the more expensive reagents or equipment. And the downsides of this is it's not as hands-on as some of the other labs can be. But I really do think that these like kind of virtual models or these simulation models are really great, especially if you pair them in conjunction with some hands-on labs. And that kind of brings me to the second solution. I can find my mouse, which is these ideas of at-home kitchen labs. So, these aren't the fancy labs that you do in the laboratory where you're transforming cells or culturing cells or making proteins, but these are labs that you can do with just, the idea of this kitchen lab where you can just use household items to be able to do some of these fun biology labs. The famous example is the DNA extraction from a strawberry, where you smush up strawberries and mix it up with some dish soap and rubbing alcohol, and you're actually able to yank out the DNA. And it's a really fun experiment to show kids, especially younger kids, this is DNA, and you find it in, yourself or strawberries, and like, what does it do? Another lab that we like talking about is the chlorophyll lab. So this is another example of an easy lab where you just go outside, pluck some leaves or grass from your backyard, and you smush it up with some acetone or some other organic solvent. And when you put it under a black light, you see the chlorophyll actually glows red. And it's a really fun discussion with your students, talking about, what is the role of chlorophyll in a plant, and why is the energy being dissipated as red under black light. So these are really these are just two examples of really fun kitchen labs where these are things that you don't have to buy fancy reagents for. These are things that you can just find around your house to be able to demonstrate some molecular biology concepts. But we're all like we're here at the SynBioBeta virtual conference. We want to think about this from like a biotechnology standpoint, right? We've made so many advances, not just in synthetic biology, but also in biotechnology to, make things like diagnostics and vaccine production even cheaper. So why not apply some of those technologies that we developed to education as well? So the first slide here I have is talking about equipment, right? As Jess alluded to, a lot of the equipment involved in molecular biology labs are really expensive. But what if we could make these pieces of equipment cheaper? What if we could it's not just the fact that they're expensive too, right? It's the fact that they're big, they're cumbersome, and frankly, they look kind of scary. What if we can make them smaller, more portable, and more user-friendly? So here's where I'm going to show off my home lab. So at Mini PCR Bio and other companies as well, there's this revolution in making smaller versions of common molecular biology equipment. So things like this right here is a gel electrophoresis unit, and you can see it's right here. It's only this big. Typically, these gel boxes cost thousands of dollars. This one only costs $350. If you want to run a PCR and amplify a gene, amplify your DNA, you typically need a really bulky, expensive thermocycler that also costs thousands of dollars. We have the mini PCR thermocycler, which is, again, handheld, very adorable. I've been running labs in my bedroom for the past couple months. Pipettes, like we have, found ways to make the pipettes cheaper and more accessible to more classrooms. And then our friends over at Oxford Nanopore, in fact, have a nanopore sequencer. So as you know, sequencers usually cost hundreds of thousands of dollars. They have a nanopore sequencer that just costs $1,000. And so like right here, what you're seeing is kind of a suite of different pieces of equipment that allows you to do things like, extract your DNA, amplify your DNA, analyze your DNA on a gel or by the sequencer. And, I can hold my entire lab up in just my two hands here and do this from the comforts of my bedroom. And the idea, you could imagine a teacher being able to do some of these labs and demo them like over webcam, like what we're doing now, and show their students, or even, maybe we can discuss ways like how do we get schools funding in order to get each student a piece of equipment so they could run this at home as well. Someone asked where the handheld PCR is from.
I'm on it, Allie. I'm gonna put the mini PCR product website in the chat.
Yes, thank you.
Yeah, for sure.
All right, and then so that's the equipment side. Let's kind of switch over to kind of the reagent side. So Jess and I developed BioBits in our PhD. So what BioBits are these reactions that are freeze-dried and cell-free protein synthesis reactions. So instead of using the cell, like E. coli cell, as the workhorse to be able to express your protein of interest, you actually can use cell-free protein synthesis, which is basically, as shown on this slide, you take the cells, you bust them open, you just take the junk that's in the inside. Well, not junk, it's very useful. Things like the ribosomes, the polymerases, and you make sure you have your nucleotides and amino acids, and then you freeze dry it so it becomes this shelf-stable reaction where it's just this little freeze-dried pellet, and then when you want to actually use it, you just add water that has DNA in it to rehydrate the reaction, and then that DNA undergoes transcription, translation, and spits out whatever protein that you were encoding for before. So this is a really easy way for students to be able to express their own proteins in a classroom without having to worry about culturing cells, without having to worry about to purify the protein. And we've paired this with our P51, which is our handheld molecular viewer. If I turn this on, you can actually see some reactions that I did last week, so they're not as bright. So with BioBits, we found that, the more bright and the more fun the output looks, the more students like it. So we actually have a lot of activities with BioBits around the idea of having students express fluorescent proteins and learning about things like the central dogma, transcription, translation, gene expression, and whatnot, just by looking at the fluorescent output here. And again, handheld, easy to use. So that's kind of the solutions from the reagent standpoint. So you have the equipment, you have, and this is just a snapshot of what is available out there. We're just kind of showing what we know from mini PCR and BioBits, but there are other companies that are doing similar things, making things more accessible. So from there, I wanted to show you really quick 2 case studies. So recently we had a lot of schools reach out to us asking us, can we do this at home? So the very first case study we did was with Professor Diana Wolf at the University of Alaska Fairbanks for her genetics class. So for her class, she wanted her students to be able to extract DNA from herbal supplements and then amplify a gene in the DNA and then analyze it by gel and then send it back to her for sequencing. And so we packed up little kits for each student. Each student got a mini PCR, a gel electrophoresis setup, pipettes, and we even sent them we have little mini centrifuges as well, so you can spin down samples. So students got all of this, and they were able to extract DNA amplify the gene, and then analyze it on gel, and then send the DNA samples back to Professor Wolf for her to do some sequencing on her end. And this was really successful. Students were able to basically complete their college project from the comforts of their own bedroom. Obviously, there were some challenges. We learned a lot from this, especially if students haven't used these equipment before. There's a lot of guidance needed, and that's really difficult to give when you're not, when you're teaching remotely. So, that could be a topic of discussion. And lastly, the second case study we did was with using BioBits at a local high school in Massachusetts, where we sent each student reagents, some BioBits, and one of our P51 viewers. And here's a screenshot of the Google Meet that the students showing off their final results, doing the central dogma lab, and they were able to learn, how DNA goes to RNA, goes to protein, and being able to visually see each step of the process along the way. And the students had a lot of fun. They commented that it was great not having just another reading or another video to watch. It was actually something active they could do. And again, we learned a lot about, the challenges of this and, what we need to do in the future. But yeah, if you're interested in either of these case studies, we actually have links down here. And Jess, if you want, or I can throw them into the chat when I get a sec.
No problem.
Yeah, you can kind of read about those. And yeah, feel free to contact us if you have any questions about if you're interested in implementing this in your own college classroom or in your own high school classroom. We have a lot of ideas and a lot of tips for getting this to work. All right, so let's have some discussion. And so the way this is going to work,
actually, if you want to pause and just ask if there are any questions. I'm answering one. Actually, I was going to type this up, but I'll just verbally answer. So there was a question about the freeze-dried cell-free extracts that we use to enable BioBits. So yes, that is the freeze-dried cell-free extract contains things like ribosomes, transcription factors, and other translational machinery that enables transcription and translation of proteins. So that in this lab is primed with DNA encoding a fluorescent protein construct. And then after incubation with the cell-free extract, that DNA is transcribed and translated into fluorescent proteins. And actually, the students can watch both transcription and translation unfold right in front of their eyes because there's a fluorescent reporter for both the mRNA that's synthesized as well as the protein itself is fluorescent.
Yeah, and there's also inquiry built in. We actually have students add antibiotic to one of the tubes, and what they see is that and the antibiotic targets the ribosome specifically, and so they see that their signal for transcription is still there, but because they added this antibiotic that kills translation, their signal for translation goes away. So it's a really great way to break down the process so they understand exactly what's going on at each process visually. All right, I think we have like 20 minutes left, so should we jump on to discussion? Does anyone have any other questions before we?
Couple more questions about the Central Dogma Lab. I'm going to put a link in the chat for more information about this now commercial lab that you can use and was used in the case study. But just some brief information. The cell extract is made from bacterial cells, which we lyse using sonication. And then the freeze-dried extracts are actually stable for long periods of time at either the refrigerator or for about 2 weeks at room temperature. That's how.
Yeah, so I'm actually linking our published papers that we published in grad school. And in here, it outlines everything. It outlines how we made the cell-free extract, outlines how we freeze-dried it, as well as example activities. So yeah, I just linked those in there. So yeah, feel free to read some and reach out and we can help you out with that as well. All right, so the way the discussion is going to work, ideally we want to have people kind of their video show up and for them to talk. We do have 30 some people, so maybe that's going to get a little cumbersome. Maybe we can try for the first few and then maybe we can just stick to the chat. And that way, if we stick to the chat, then more people can talk. Versus just going one by one video. But maybe we'll just start here. So, the first question I wanted to ask is how do we get students excited about molecular and synthetic biology? And kind of a sub question of this, what are your fondest memories of learning biology in the classroom? So I guess for this question, maybe people can type up in the chat, if you're an educator, what ideas you have about getting kids excited. And because right now, as I feel like a lot of teachers and educators can comment on this, student motivation's at an all-time low right now. With everything that's going on, the last thing students want to do is learn, unfortunately. And so how can we motivate them? How can we keep them excited through this time, even if they're learning at home? Because, we still want students to get interested in science because we do need to build our SynBio workforce in the future.
As some of the comments in the chat are coming in, I'll just kick us off by saying that I think hands-on activities are a great first step. And I know that hands-on experimentation is part of what I love about science and why I went through a PhD and now to a postdoc and, beyond. But I think I've had success in some of the teaching and outreach activities that I've done in linking experiments to concepts or current events that they might be hearing about on the news, like CRISPR, genetically modified organisms, helping students put scientific context behind some of the things they may be hearing about in the press.
Yeah, for sure.
Hopefully that gave us enough time to everyone enough time to think about their own experiences. I see someone seconded my thought that applications to real world situations are really important as well. Meaningful experiments.
Yeah, for sure. I remember the example I love giving is like the first time I dissected a fetal pig in biology. And that's not molecular biology, but just like being able to get hands on and actually see the organs. I was like, this is it. This is what I want to do. I love biology.
Seeing cells divide. That was a similar theme. Punnett squares. That's a throwback.
Yeah, if anyone's playing Animal Crossing right now, I've been drawing Punnett squares to figure out how to breed my flowers in that game. And so I'm applying what I've learned in molecular biology to that. Okay, so
yeah, let's keep going, and then maybe at the end we can open things up based on more responses.
Yeah. So we showed a couple examples of how we can make these technologies and equipment and resources more accessible to everyone, right? We showed you how you can go from like $100,000 sequencer or like a couple $1,000 thermocycler down to just a couple $100 thermocycler. But a couple $100 is still quite a lot for some schools, right? So are there other SynBio or biotechnologies out there that can be applied to education? Are there strategies we can employ to get these very accessible hands-on equipment into the hands of students? So that's kind of a very broad question. We've already made this very accessible and very cheap. How do we kind of cross that last barrier and get these in the hands to the homes of students during this time?
I don't really have an idea for technologies per se, but when I saw this question, I thought that, while we're all isolated at home, it has obviously created a lot of challenges for hands-on education, but what it has kind of brought to the forefront is opportunities for virtual lectures that can be recorded and can reach a really broad audience. I know that during quarantine, I personally have been seeking out a lot of virtual seminars and virtual conferences in areas that I may not normally look to, just to expand my knowledge, particularly anything related to COVID-19. I've been paying very close attention to, and I think probably many students interested in science are doing the same. So all that to say, I think that we can leverage the virtual environment that we're in right now to really take the time to do some science communication and reach a really broad audience with some of these scientific ideas.
Yeah, for sure. Kind of another strategy I just thought of, and this is something I mentioned earlier in Natalie Kudel's session that she did, kind of talking about the idea of scientific programming in these days. The idea of industry, biotech industry pitching in, and either providing loans of pipettes, providing funding, to cover shipping costs, to ship, some of these equipment to students, that kind of thing. Because, these are the students who are they're going to grow up and be the ones who are going to be the scientists of the future. So it's really worthwhile, to invest into these students. So, industry with their resources could be a potential area where we look into to kind of get funding to subsidize some of this for students, especially for schools, really, like you say, like rich private schools probably could afford this, like one mini PCR for each student. But, then we're not targeting the demographics that we want to target. So, the idea of like making this more accessible for kind of every student, regardless of their like socioeconomic, their school background, to be able to get access to these kind of things. Someone asked, where are we located? So Mini PCR and BioBits, which is at Mini PCR now, is located right outside of Boston. So we're in the Boston area, but we do ship all around the world. Just a quick humble brag. We actually have one of these on the International Space Station. So we even ship to space. So we can definitely ship to wherever you are because we can ship to space. Yeah.
Great.
And I'll just throw the last question on here. Maybe we can have a more holistic discussion for the last 10 minutes. And the last question kind of ties into what we were saying before. How do we bring these hands-on biology activities to students in our current hands-off world? So, if there's teacher and educators out there right now and parents, like how have you been teaching your students and children, and like what are challenges we need to overcome in order to get these things into the hands of students?
Tom has a nice thought in the chat about linking students to online resources from SynBioBeta or IndieBio for applied examples of synthetic biology, and then following up perhaps when we're back in person with hands-on activities from BioBuilder and hopefully from Mini PCR and BioBits as well.
Mm-hmm. Hello.
Hello. I thought I'd offer to jump in here. I am formerly from SynBioBeta, helped scale that a while back, and am now working for an organization called Focus Bio, where we are trying to establish synthetic biology education and outreach in biobelt communities. And that's why I asked where you were located. I've been doing this for about 2 years, and I will tell you the biggest problem we have isn't necessarily getting the students interested, it's getting buy-in from the administration and the teachers, because they don't know what they don't know. And two things or two strategies that have worked really well here, and I'd love to have a conversation offline with you.
Yeah, for sure.
Two strategies that seem to work are, one, a teach the teacher model where we go in and we run student programs. And we've done this in a webinar as well as in the classroom pre-COVID. But the first year, the first semester we run it, we teach the student while the teacher observes. The second time we co-teach, the third time they teach, we observe. And so by the third semester, the teacher is running the program on their own. But a lot of even the high school science teachers, I realize Silicon Valley and Boston are whole other worlds where many of your parents do work in biotech or have at least more than a high school education. Here, our science teachers are lost when we start trying to teach them. So really doing that teach the teacher model is a huge advantage. The other thing is to piggyback it on tech education. And we've had to go back, and even before the strawberry DNA extraction, we've had to do the gumdrop in the model. And we've done the forensics where we match up the DNA pattern to the person. So they get the concept of DNA as a code. And we find that when we do that first, it really helps a lot. And then the students that, have been doing this, we also tied it in with a partner organization that does coding and computer classes. And to them, it was just almost like one more language then. So those two things have been really successful and may help.
I will
I did notice.
Sorry. Go ahead, Allie.
I was going to say, I did know that someone did mention bioinformatics as a really great way. It kind of solves a lot of issues with, you don't need reagents, and there's a lot of free bioinformatics tools like BLAST out there that people could use. And so, yeah, that's a really great point. Sorry, Jess.
Yeah, I also just wanted to say that Allie and I both have a lot of experience with the teach the teacher model.
Oh, yeah.
So just because I'm in Silicon Valley now and Allie's in Boston now doesn't mean that we don't understand the challenges that teachers face in kind of onboarding these things. You know, I'm from the Midwest, so is Allie. Not every school that I went to had a lot of resources. So we fully appreciate, and I've worked with training teachers during my PhD. Allie is currently training teachers through her work at Mini PCR. So yeah, we totally appreciate that. And we're making opportunities for teachers to get acquainted with the products that we've talked about today, at least the ones that are housed within Mini PCR. So if anyone in the chat, anyone in this group is interested in learning more about these hands-on resources, definitely get in touch. We have an online demo actually of one of the BioBits labs, as well as Mini PCR is continuing to roll out online demos of their labs to help bring this training in an online era.
Yeah, and just to put it aside, like normally before the pandemic, we would travel to a lot of schools, especially schools in more rural areas like where you're from, and do teach-to-teacher workshops where we train teachers how to use our equipment so they can do it with their students as well. We also have a loaner program, in fact, that we actually send out our equipment for free as a 2-week loan to students and teachers so they can actually, use our equipment for a couple of weeks before returning. So that's a, good way for them to get exposure without, the huge upfront costs. So yeah, if you're interested in that, I put my email in the chat. Feel free to reach out to me. We are doing whatever we can right now to kind of support teachers. And we found that especially in this kind of current circumstance, teaching to teachers over webinars or over chat. I'm doing a lot of teacher training workshops over this next coming summer. And so, yeah, feel free to reach out, Kate, or anyone else in the chat who's interested in kind of networking with us and, getting some teacher training in.
In the last 5 minutes, because there's been a lot of interest in bioinformatics activities, is there anyone on who has experience running that kind of an activity in the classroom and would want to share their experience? Allie, it looks like Todd is willing to talk about this if you want to bring them on.
Oh, yes. Yeah. So, a couple with both teachers and students, but we have, I like to call it the world famous blast tutorial. If you type blast.
Oh no,
we lost Todd. Hopefully we can get him back. I will just mention that in some of our published papers on BioBits, these aren't. Oh yeah, go ahead.
Oh, okay. Yeah. So so a couple experiences. I typed it in. One is just go to a website that does a 6-frame translation. It's a really simple thing to do, and you can do it with biology teachers. And if they've never done this before, the first question they're going to ask is, why are there 6 protein sequences here? And that's a great place to introduce the whole concept of what you can do with bioinformatics in the computer, because what you're going to see on the screen is really expressing what we're teaching in biology. We teach about reading frames, we teach about these things, but until you see a computer give you that data, the connection that people will make. And then from there you can go and do, blast kinds of experiments. Classic is, compare hippopotamuses to dolphins and things like that, because, they have, they're biological, but they're unexpected results from what people have conceptualized. So whenever you're thinking of how to do that, you want to come up with things that kind of break those barriers. Does that make sense? Make teaching moments. Yeah.
For sure.
Before we got you back, Todd, I was just going to mention that in one of our published papers on BioBits, we have a similar kind of blast-related activity related to gene editing and CRISPR that could be useful for people. So I'm going to post the link to our open access manuscript in the chat. And if you go into the supplement, there are curriculum resources that we developed that might be useful. So I'll put those up.
Yeah. And just because we have like 3 minutes left, and I saw some people mention this in the chat, maybe the last thing we can discuss about is obviously the major barrier to this is cost. And obviously bioinformatics is a way to get around that cost. But if you do want to get, this equipment and these reagents into the classroom or into the homes of students in this current time, does anyone out there have any ideas or thoughts about, how do we kind of circumvent the idea of like how costly this is? Like, are there funders that we go to? Are there industry contacts that we leverage? Like, what are people's strategies with dealing with cost? I guess it's a big problem. Yeah, I don't know if there's anyone here from industry who could speak on this, or anyone who runs a program who can speak on what has been successful for them to fundraise, or anything like that.
I haven't had a lot of success with industry. We've had a few small, sponsor a classroom kind of thing. And generally, I think a lot of industry partners have a hard time bringing it down to a middle school or a high school level. They're much more concerned about workforce development, which is understandable. But I think the one place we've gotten some better results is from some nonprofit organizations. One has a nice endowment, and they've been very helpful in supporting a number of our programs. So maybe more nonprofit foundations than industry.
Yeah, for sure. All right, for our last minute, does anyone else have any last minute things they want to get off their chest? So I think this is, just to wrap up, I think this has been a great discussion. I love hearing, I'm very in the molecular biology world, but I love hearing the things about, the advances in bioinformatics and leveraging those tools to be able to use for students, the ideas of like virtual simulations, that kind of thing. I feel like combining all of this together might be, the way to go. A little bit of everything for students so they get a really comprehensive view of what molecular biology and what synthetic biology really is.
Thank you both. This was an amazing presentation.
Thank you all for the hard work that you're doing to educate students during a pandemic.
Yeah, it's definitely very impressive, the amount of effort and energy you guys need to have to, motivate students to do anything right now.
All right, well, I think we can leave it there. I'll just say that both Ali and I are happy to connect with any of you if you have additional questions. Hopefully some of these resources that we've shared are helpful, and thanks for being here.
Yeah, thank you. All right, do we get kicked out at a certain time, or do we just go whenever?
I'll hang out. How about we hang out for a few more minutes in case there are any stragglers, any lingering questions. But I think then we can probably call it.
Yeah, thank you. Yep. So nice to see everyone here talking. Thank you, Tom. Thank you, Todd.
Yes, thanks for everyone who chimed in, whether in the chat or via video. That was a really great discussion.
Yeah, and again, I linked some of my contact info. You can also find us both on LinkedIn. Feel free to reach out to us and talk further offline about how we can help support your programs, your classroom, your child with all this learning about synthetic biology and molecular biology. So,
did you share maybe like a general mini PCR contact email in case they're interested in any of the resources that we shared?
Yeah, they can contact team at minipcr.com. I'll put that in the chat.
We have a question. Have you patented this kit? No.
Some aspects are protected. Yeah.
Oh well, you mean like the viewer?
Mm-hmm.
I see.
Yeah.
But the BioBits reagents in particular are not patented, and in fact, there are ways if you have more sophisticated lab equipment, there's ways to try to recreate it in yourself. And we detail all that in our open access manuscripts, some of which I've posted here.
All right, I already have some LinkedIn networks.
Oh, really?
Uh-huh.
That's good. Okay, we still have some people on, so maybe we can just stay for a couple more minutes, but then we can probably take off. That was great.
Mm-hmm. Yeah, that was a lot of fun. All right, bye. Say bye to the chat. All right. Let's see if I can look up some people. All right, we'll probably just give it another minute. If no one's going to say anything, we can hop off and hopefully, hopefully we started some fun conversations offline.
Definitely.
Mm-hmm.
Yeah, I wish there had been more time for discussion. We barely scratched the surface, but it's how it goes, I guess.
Yes, too much to talk about. All right, I think we could probably leave now.
All right, we'll leave it there.
Oh, did we record this meeting? It says recording. It says the session is being recorded, so I'm assuming SynBioBeta is going to be posting these somewhere after the fact. Yeah, I hope so. Yeah. So yeah, I think all the sessions from today are being recorded.
Great. All right, I think our audience is quickly dwindling, so we can probably take off. Thanks, everyone.